Well, our large intestine is here. And large intestine consists of how many parts?
4, 5.
3 parts. Large intestine consists of 3 parts. First part, the smallest part, this one, and this one, there is cecum. So our ileum empties into large intestine in the region of cecum. All part of large intestine which is below this junction is called cecum, and this vermiform process will also start from it. Second part is the longest. It goes like that. And this part is called colon. But colon is subdivided into 4 parts. First part, from this junction, ileocecal junction, which goes upward, is called colon ascendens. And borderline or borders of this colon ascendens, distal border is ileocecal junction, but proximal borderline is flexura, right colic flexura. Then second part of colon is this one, this transverse colon, which has borders. Right border is right colic flexura and left border is left colic flexura. Next, third part of colon is descending colon, which goes from left colic flexura up to level of crista iliaca, iliac crest, this bone. And approximately at this level is crista iliaca. And next part, this one, is sigmoid colon, which is last, fourth part of the colon. And then at the level of promontorium, I hope you remember what is promontorium. Do you remember what is promontorium?
The curve on the iliac crest?
No.
No, that is in the hip bone,
No, not hip bone. Promontorium can be found on the vertebral column. You're right, this is between sacrum, base of sacrum, so means between first sacral vertebra and last lumbar vertebra. This elevation forward into the cavity of the pelvis. And this promontorium is borderline between colon, if more exactly between sigmoid and rectum. So there are 3 parts of large intestine: cecum, colon, and last one is rectum. There is rectum. Our rectum has two divisions. First, anatomical division, and second, surgical division. Look, in rectum we have dilated part, this one, which is called ampulla recti. And this ampulla can help us to divide rectum. According to surgery, there are 5 parts of rectum, 5 parts. First part lies just above this ampulla, this one. This part is called pars supra-ampullaris, because it be above the ampulla. Then ampulla proper is subdivided into 3 regions, 3. First one, upper part, is called superoampular part, then mid-ampular part, and inferoampular part. And last one, the lowest part, this one and this one, is called canalis analis. And canalis analis terminates by anus. Anus is opening, and this opening is closed, and it is closed by 2 sphincters. 2 sphincters are here near anus. First sphincter is this one, and it is called musculus sphincter ani internus, internal sphincter of anus. And this sphincter is involuntary. You cannot control it. And musculus sphincter ani externus, which is voluntary, and you can control this sphincter. In the region of canalis analis, you can see longitudinal folds. They are very large, and because of this, they are called columna analis. Not folds, but columns. There are spaces, there are depressions between them, which are called sinus analis. And these sinuses are filled by mucus, with the help of which feces can pass through it very easily. Well, in this region, in the region of columna, inside, in this wall of the rectum, we can find special veins, which are called hemorrhoidal veins. And if a person is sitting very long period, these hemorrhoidal veins enlarge and hemorrhoid is occur. Well, that's why this specific feature of mucus of the large intestine of rectum, that pars analis, canalis analis, has longitudinal folds, which are called columna, but rest part has semilunar folds. Semilunar is this like type of transverse folds. Well, now let's talk about differences between large intestine and small intestine. All differences between them I can divide into two parts, two groups. External differences, when I cut the skin and look at this preparation or look at the human body, I can differentiate where is the large intestine, where is the small one. And internal, when I cut intestine or during endoscopy, I have to know this one is large intestine or small intestine, what are differences in mucus. So now I will explain only external differences. There are three basic external differences. Three. First one, large intestine has processes, this one. These processes are called appendices epiploids. They are special processes which contain fatty tissue. You can compare with small intestine, no any appendices. Only in the large intestine. They are clearly visible. When we will have COPs, you will see them. Then, second very important difference, presence of tenia. Look, this line is called tenia, this one and this one here. What is tenia? Tenia is muscular cord. Muscular cord of large intestine has same structure like small intestine. It has two layers, circular and longitudinal layer. But longitudinal layer is submitted by only 3 strips. These strips are called tenia. So there are 3 tenia. So please remember that in this part of the large intestine, here, there is only one layer, only circle layer of muscular cord. Longitudinal layer is submitted by 3 tenia only. Understood this? These tenia are shorter than length of whole large intestine. So large intestine proper is longer than this tenia. That's why because these tenia are shorter, all large intestine makes circulation. This circulation, this one, are called haustre. And these haustre are very important for digestion of plants. That's all. Internal differences, you'll prepare yourself.
Spleen.
Spleen is here. Look, and duodenum, this one, passes around the head of pancreas. Today you don't know pancreas, but from one side of pancreas is duodenum, but on another side is spleen. And now come to the preparations and I will show you something. No, before spleen, what can we see here? This part of large intestine, this ileum, ileocecal junction is here, vermiform process. So this junction is borderline between cecum with vermiform process and ascending colon. And now from outside you can see these processes. So how these processes are called? Do you remember? Appendices epiploids. So this one, small intestine, it has no any processes. This, a lot of these processes. Well, now spleen. So look, this is spleen, and first of all, we can recognize correct anatomical position of this body, of this organ. So how can I do this? Remember our class. I said if organ is paired, we must find how many features? 3 features. If organ is unpaired, how many? 2. Spleen is paired or unpaired? Unpaired. That's why there are 2 features will be enough to distinguish. First of all, look at this spleen and we can see one surface, another surface. One surface is convex and almost smooth. It is called facies diaphragmalis. Usually, facies diaphragmalis should faces to the lateral side. So I can put this organ like this, no more. Another surface, this one, is irregular in shape because it comes in contact with internal organs. That's why this surface is called facies visceralis. So 2 surfaces, facies diaphragmalis, which is convex and smooth. This one, facies visceralis, is irregular. Facies visceralis contains place where blood vessels enter our spleen. This place is called hilum or porta lienalis. Porta lienalis. Well, what is the second feature? Now I can look at the edges. Look, one edge is convex and smooth, but another edge also is convex, but it has incisura. They are not clearly visible because this organ is damaged, but here some incisura, 2, 3, no more. So edge which contain incisura, notches, is superior, margo superior. Here is margo inferior. And now two features are enough. I know that convex smooth surface faces to the lateral side, and superior edge has has incisura. There are no incisura here. So this is correct localization of our spleen. And now if you know that this is correct localization, now you can distinguish two poles, two ends. One end is extremitas anterior, another is extremitas posterior. Anterior end and posterior end of spleen. That's all. No need to show something else.